All right, so today we're going to talk about how to pull a blood sample from a cow. And there's really two options that you can use. You can pull from the jugular vein or from the tail vein. For the most part, if you don't need a large volume of blood, pulling from the tail vein, or what is commonly known as the coccygeal vein, is going to be the better option. So today I'm going to walk you through what tools you'll need in order to pull that blood sample, as well as how we actually perform that blood sampling technique. So I'm going to walk you through some of the tools that you'll need, and then we'll actually use some of the ladies behind me as an example on how to pull tail blood. So first off, you're going to need gloves, and we'll put these gloves on before we actually go to the cow. Not that you should come in any contact with any biological samples, it's just always a good idea to wear gloves whenever we're working with cattle. The other thing that we will need are what we call blood collection tubes, or our vacutainer tubes. And these are unique tubes, and I know a lot of people like to pull blood with a needle and a syringe. I think this is just the simpler method by which to pull that sample, and that's because that tube already has a vacuum within it. So once you actually puncture the top of the tube, it's going to want to pull that sample down into the tube, so it makes it a little bit easier of a sampling technique. So again, these are called vacutainer tubes. They do come in a variety of different kinds, or ones with different additives within them. Each different kind of additive will have a different color top to the tube. For most samples that you would take, a red top tube, or one that has a red stopper, no additives, is going to be perfectly fine. And so this would be the type of tube that you would pull most commonly if you were going to maybe want to preg check your cow using a biofrin test. You could use a simple red top tube. So after you have your vacutainer tube, the next thing that you're going to need is what we call the vacutainer needle. And again, much like our tubes, our needles can come in a little bit different styles. What I have right here is the Eclipse vacutainer needle. This is a 21 gauge needle, 1.25 inch in length. You can see some of those needles go down to an 18 gauge. Some of the advantages of maybe going to the 18 gauge, which has that larger bore or diameter to the needle, is it is a little bit more sturdy when you're pulling that blood sample. In addition, you'll have less hemolysis or rupturing of your red blood cells. So a lot of times, if you need a cleaner sample, those larger bore needles will work more effectively. For the sample that we're going to pull today, and for most samples that I use, I find that a 21 gauge needle works just fine for my sampling technique. So again, this is a 21 gauge needle. I will show you the parts of this needle. So this is the actual puncturing portion of the needle, or the portion that will actually insert into the cow's tail. You'll notice this clip here. This is for safety reasons when you get done pulling your sample, to be able to clip that over the needle so you don't risk injuring yourself. On the other side of this needle, so it is a double-sided needle, you will see that we have another needle, but this end is actually covered in a nice rubber piece. What that allows to happen is for that blood not to come through this needle and begin to evacuate until you're ready for it. So this end will actually end up puncturing that red top in your vacutainer tube. And so when you make that pressure, and I can go ahead and do it on this one, when you make that pressure and actually puncture the top of that vacutainer tube, we'll see that the end of that needle is then exposed, which then allows that sample to be collected when you're ready and when you've actually tapped into that particular tube. So again, that side of the needle will go into your tube. The needle end that is fully exposed will go into the cow's tail. When you've completed your sampling, you can simply pop that Eclipse over your needle for safety, so you don't risk injuring yourself when you're done collecting the sample. Now, there is one other key piece of equipment that you're going to need to make sure that this sampling goes as simple as possible, and that is the blood collection cuff. And this cuff is what's going to make it a simple assembly between your needle and then your blood collection tube. So in order to use the cuff, we'll take another one of our needles out. We're going to take off the white end of this vacutainer needle, which again exposes the end will go into the tube. That then is going to simply screw down onto our cuff, And what this allows us to do is have a nice stable apparatus for the blood collection exercise. So we don't have to worry about stabilizing our needle, that cup, or our tube. That cuff is going to help do that for us. All right? So again, this is what it will look like. This uncapped will be in your cow's tail. Once you're ready and feel like you have the correct placement, we will then use this cuff and tube to then begin that collection process. So now that we know the parts and the pieces necessary for blood collection from the tail or coccygeal vein, let's go to the cows and actually see it in practice. So before you actually go to pull your blood sample, you actually need to know a little bit about tail anatomy. So again, we're doing a coccygeal venipuncture or tail bleeding in this cow. And the reason it's called coccygeal venipuncture is not only are you tapping into the coccygeal vein, but you're also going to be paying specific attention to the coccygeal vertebra, which are the actual vertebra that comprise this animal's tail. And so here are some key aspects. You're going to be able to feel these vertebrae as we move down the tail. A lot of times once we pick the tail up, some of those are easier to see. So you'll be able to feel the bony-like structure of that vertebra. So here, this is Kelly. We can feel that coccygeal vertebra here. So a bony-like structure, and then it will feel more tissue-like or meaty, if you will. And then back to a bony-like structure. So there is her next vertebra. Again, tissue or meaty, back to another vertebra, so on and so forth down the length of that tail. So one of the key aspects is that we want to make sure that we collect our sample in between those vertebrae or into that meaty tissue-like segment, not into the actual vertebra. The other thing about the tail is that coccygeal vein sits almost midline within the tail in an indentation. Depending on the body condition score of the cow, it's going to be how easily we can actually feel that vein or that indentation in which that vein will sit. So it varies a lot on Kelly here, who's a little bit thinner or lower body condition score. We should be able to feel that quite easily, which is one reason that she was selected for this demonstration. The other thing is because Kelly is white, we should be able to see some of these vertebrae a little bit easier when we pick up the tail. So this tail sample will be collected from the underside of the tail. So I'm going to pick the tail up here in just a second. From there, we're going to look a little bit more at anatomy and discuss exactly where we'd want to pull our tail sample from. So if we take this into the tail, and we're literally going to pull it straight up, so we can see the underneath portion of Kelly's tail. Here, hopefully you can appreciate, you can actually visually see that vertebra, that coccygeal vertebra that sits right here in Kelly's tail. Again, a meaty segment, then another vertebra, meaty segment, another vertebra. And you can actually, if you look down up here at this portion of the tail, you can almost see that midline indentation that I was discussing, which is where that coccygeal vein will actually sit. So if you think about anatomically where we're looking to collect our sample from, it is going to be between these vertebra, between the vertebra, and directly midline into the tail, and we will go in at a perpendicular angle to that tail. So again, between these vertebra, midline, perpendicular into that tail. The other thing is you might ask is where on the tail do we collect that sample from? If you go further out the tail, one of the advantages is if for whatever reason you may be below that vein, you're able to work your way back towards the base of the tail. However, a lot of times it's harder to get a nice decent blood flow out there for blood collection. So I tend to like to stick closer to the base of the tail, somewhere in this region, for that initial blood collection sample. So now what we're going to do is get our supplies ready and collect our first blood sample completely and fully from Kelly. All right, so now we're actually going to collect our blood sample. You can see here that I have on a pair of bibs, which makes it nice because I can put all my tools in here that I'm going to need. If you don't, maybe having a table or another set of hands would be mighty useful because it is going to involve a lot of different moving pieces, if you will, to collect the sample. So again, I'm going to have my needle first, and I'm going to be attaching it to my cuff. So again, I'm going to take off the white end of that needle, which is going to expose the end of the needle that is covered in that rubber piece. So that's going to be what actually is going to go into my blood tube. I'm going to take that cuff and we're going to screw that onto there. All right. So at this point, my apparatus is ready for blood collection. This is going to contain the needle that will actually go into Kelly's tail. You want to make sure that you have your blood tubes ready to go for this. So what I'm going to do now is I'm going to lift Kelly's tail. I'm going to simply clean the area with some alcohol here that I plan to collect my sample from to make sure that it's nice and clean. You're then going to see me palpate for where those costigil vertebrae are, as well as that midline indentation that I'm going for in order to collect my sample. You're then going to see me insert the needle. I'm going to do that fairly quickly and fully, and then I'm going to release this cuff and needle. I do that quite purposefully. I'm going to release it because if Kelly then wiggles, it allows that needle and cuff to stay with her. If I try to hang on to it, even if I try to be flexible and moving with her, most often what happens is I end up pulling it straight back out. So again, I'm going to insert that needle, release it until she has a second to stop wiggling, or maybe just stepping back and forth before I then go to attach my tube. All right. So again, we're going to put this tail up. I'm going to look to collect my sample somewhere in this general location. We'll palpate that again here in just a second. Make sure it's nice and clean so we can see this vertebra here. Costigil vertebra, nice meaty section. We can even see that midline indentation. Next vertebra here. All right, so again, make sure that's nice and clean. We're going to take the cap off and expose the end of that needle. And again, you're going to watch me insert, so midline, perpendicular. I often use my finger against the tail to help brace that. Insert and release, right? So that gives her a second to move around, if you will. Then I'm going to take this, and it takes a little bit of pressure to be able to push that up. And then you can see that collection. This vacuum, again, within that vacutainer tube is naturally pulling that blood out. It will reach a full point and will stop pulling blood from that tail. And I just generally support it lightly in case she does wiggle. So here, hopefully, can you see that is a full tube. Now, for the animal's sake, what I do next is I'm going to take that tube off. And I'm going to give that an opportunity to sit. The whole purpose of giving it an opportunity to sit is to allow that to clot. That'll help for when I pull that needle out that I don't end up with a stream of blood or a hematoma forming on that animal. So then sometimes I will gradually move that needle out. Again, giving it the opportunity to clot, especially if you need to collect multiple blood samples. That would be important so that you didn't get hematoma formation. And then you can see I've pulled that needle. And just that few minutes of extra time, really seconds of extra time, make sure that that's a nice clean removal of the needle and we don't have any further blood expression. So that's the collection from Kelly here. We're going to do one more as a demonstration for you on the full collection process. All right, so here's another example of how to pull costigil, venipuncture, or tail blood from a cow. I'm going to walk through the entire process again, showing you kind of anatomically where I would pull on this cow, and then again how we would ultimately pull that sample. So this is Cheerio here. So again, we're going to lift this tail. We can see the vertebra nice here on Cheerio. So one here, and again, one here. So again, we can see those quite easily on this animal. If you were to get an animal, so we can visualize them on her, if you were to get an animal that's more heavier conditioned, beef cattle as an example, palpating for those vertebrae will become essential. And so that's going to be a technique that you'll use is palpating. I generally palpate from the exterior of the tail just to get a general idea on where those vertebrae are. And then when I lift that tail, I'm going to run my finger up and down in order to feel those vertebrae. And again, it's kind of cheating here on Cheerio because I can see them quite easily. But again, on a cow that's carrying more condition, you will also be able to palpate or feel where those vertebrae are located. So I'm going to go ahead and get my items ready here. So again, we're going to take this end off. That's going to be the one that goes inside of my cuff. Right, my apparatus is now ready. I have my blood tubes in my pouch here, ready to go. So you need to have those easily accessible to you. lift her tail. I am going to clean the segment that I think I'm going to use. So in Cheerio, I'm going to go between this vertebra and this one. Again, we're going to be working off of midline on that animal. So take that needle end off. Palpate for where that indentation is on the center. Again, use my finger to brace. Simple and release. Make sure she's not going to wiggle. Take my tube, place it on. In this case, I didn't immediately get blood, so I might need to move it just gently until I get a nice steady stream. And again, maybe not as steady as we would like it initially. So you do some slight manipulations. Being very gentle, you got to think that you're dealing with a relatively small structure, so big movements are not necessarily required. The other thing that I would say, and a good thing to point out as this blood is coming, is that if she were to wiggle and this entire structure, including the needle came out of her tail, that vacutainer tube would then be considered blown, which means that you would lose that vacuum on that tube. And if you needed more sample, you would need to get a new tube for that. So do realize that anytime that needle is not in her tail, but the other end is within that tube, you remove the vacuum on that tube. So you'll actually need to get a new tube out to collect any more of that sample. So again, just taking an opportunity here to let Cheerio clot. Again, just for her sake and her benefit, so we don't have any excess blood once we're done with our collection. Okay. So again, this has been a very quick demonstration on both how and where you would collect coccygeal or tail vein blood from a cow.